Today we're looking at the process of muscle contraction, which will include the sliding filament theory. And this starts with nerve transmission. So performers have receptor neurons which receive information from their environment, such as stimuli. These receptors send the information to the brain via the central nervous system. The brain then decides how to respond to the stimuli in the environment, and these impulses from the brain are sent through the central nervous system to motor neurons, and motor neurons receive the impulse and make the relevant muscles contract. They do this by sending ACH across the chemical synapse caused by the contact between a motor neuron and muscle fiber, and the ACH bonds with the receptors on the other sides and then travels through T tubules. So this works with the sliding filament theory, and sliding filament theory explains how the filaments of actin and myosin work together to cause contraction of the muscles. The myosin head attaches to the actin filament to create a cross bridge, and the myosin then pulls actin towards the A line before releasing the actin filament and bonding to another actin filament, repeating the process. So here you can see that the myosin filament is the red filament in the middle, and the actin filament is the green filament on the outside, which is pulled towards the H zone, and the Z line is therefore pulled closer to the H zone when contraction occurs. So here the cricket ball would be the stimulus, the eyes would be the receptor neurons, and the brain would receive the information that a cricket ball is in the air, and then would choose what to do and send the impulse to the motor neurons. The motor neurons would then send impulses across the neuromuscular junction in the form of ACH, which would be received by the receptor sites. The receptor sites would then send the impulse through the T tubules, which you can see on the right here. And also, this would stimulate the release of calcium from the sarcoplasmic reticulum. As you can also see here, that this is the sarcomere. So the myosin is the red filament in the middle, and the actin is the blue filament on the outside. And the this line here is the Z line, which is pulled closer to the middle when contraction occurs. So the muscle shortens and creates movement. So here you can see that there is troponin and tropomyosin on the actin filament, whereas the myosin filament has the head coming out here with ADP and an extra phosphate molecule. So this combines to create ATP, which means that the myosin has a store of energy and is ready to cause contraction. But as you can see, the binding site on the actin filament is blocked by the troponin and the tropomyosin. So the calcium that was released from the sarcoplasmic reticulum bonds with the troponin and pulls the tropomyosin away. So the bonding site then becomes available for the myosin head to attach to. And here you can see the top part of the diagram is the relaxation phase of the muscles before the myosin head has pulled on the actin filament. And then you can see when the myosin head's bonded with the actin and pulled, then the Z line has been pulled closer to the middle. This occurred because the bonding site has been released and a cross bridge has been formed, and then the ATP that was stored in the myosin head has been broken down by ATPase to provide the myosin head with energy, which then pulls the actin towards the midline, which is called the power stroke, which is where the muscle actually contracts and shortens, which will create movement. And then this process can't occur again because the myosin and actin stay together until more ATP reaches the myosin and releases the bonding site there, the cross bridge. So when ATP reaches the myosin releases the actin bonding site, the troponin and tropomyosin go back into place and cover the bonding site, and so more calcium is needed to make the bonding site available again. So unless more impulses are sent to the muscles, then the bonding site is unreachable for the myosin, so no further contractions can occur without further impulses in the brain. So once the action has been completed and impulses stop coming to a performer, so in the example we were using, once the cricket ball has been caught, impulses will stop coming, calcium will stop being secreted, and the bonding site will be covered, so the myosin could no longer bond with the actin filament, and the muscles then return to resting length. But if the impulses kept coming and the ball had not been caught yet, then the muscles would keep contracting until they're at the appropriate length.